Okay, so welcome for another installment of Polish NMR seminars. This is a recording after the seminar because of my error in the setting up the recording. So we're recording first 3 slides of Aleksandra, and this would be published on YouTube together with the rest live slides. So sorry for this problem, and let's invite Aleksandra Ptaszek from University of Vienna. Her talk will be bringing computational NMR approaches to unravel protein-ligand interactions. So I'm stopped sharing the screen, and the screen is yours, Aleksandra, and sorry for the problem.
No problem. Just tell me when to stop.
On the first slide.
Okay, so thank you very much for the introduction. And today I would like to present my PhD thesis that I defended a year ago at the University of Vienna. And let me start with a short background. So my PhD work focused mainly on NMR chemical shifts that, as probably this audience knows, are the main observables of NMR spectroscopy. And depending on the local environment, each nucleus in the molecule has a characteristic resonance frequency that can be detected by NMR. And chemists are able to assign these signals to specific atoms in molecules. And this is the basis for extracting structural information from the NMR data. And they find NMR chemical shifts find many applications, not only for identifying small molecules, but also for studying biomolecules that are the main focus of my thesis. And the first application for chemical shifts in proteins is secondary structure identification by comparing backbone chemical shifts with random coils. We can also identify conformational changes using carbon side chain chemical shifts. For example, here the conformation of isoleucines. And chemical shifts are not only sensitive to intramolecular changes, conformational changes, but also to intermolecular interactions. So you can see here how isoleucine interacts with the ligand, and we can see a chemical shift perturbation caused by the interactions of the methylene proton with the aromatic ring. And of the high importance is also the fact that NMR can be applied on the samples in solution state, which makes it perfectly suited to study biological systems. So compared to X-ray structure, the NMR can provide the information about proteins flexibility. However, to extract structural information from NMR spectra, especially in case of proteins, it is essential to map peaks from the NMR spectra to the molecule. However, proteins NMR spectra are extremely complex, especially when the size of the protein grows. So the spectra are very crowded and hard to interpret manually. And this is where computational tools become important. And they can assist in peaks assignments. They can be used to validate experimental structures, typically in organic chemistry, and they can even help to refine structural models, which you will see today during the presentation. And computational methods for chemical shifts predictions fall into two broad categories. The first is ab initio methods, which are based on quantum mechanics, And here we calculate chemical shifts from first principles, and there are also data-driven methods, such as machine learning methods that learn from experimental and ab initio data sets. And my work focused on combining NMR data with computational tools to bridge the gap between static structural data and dynamic molecular behavior, to contribute to progress in structural biology and to support drug discovery efforts. And this is the outline for today. So I will present 3 papers. The first 2 are focused on proton chemical shifts in protein-ligand systems using quantum chemistry methods. And the last one is machine learning predictor for protein side chain chemical shifts. And here will be also a little follow-up with the application of UCB shift for metal assignment. So let me start with the first project, where we used ligand proton chemical shifts in order to understand protein-ligand binding mode. The system of study that we chose was the nanomolar binder bound to NSD3 PWWP1 domain. And the standard procedure when using NMR shifts for protein-ligand systems is using backbone amide groups, a chemical shift perturbation. However, in this case, we only have 2 backbone amide groups that are within 4 angstroms from the ligand, so the rest will not change, they will not be affected by interactions with the ligands. So this will not give us a lot of information about the interactions in the system. And there are also 37 carbon-bound protons in the binding pockets. However, this requires side chain assignment, and there are mainly methylene protons, which are hard to interpret because of peak broadening. So instead of that, we look at ligand chemical shifts that were measured by Gerald. And we look at not exchangeable protons, so only carbon-bound protons in this case. And so this is the experimental information that I used. Chemical shift of the protons in the bound form. And my first question at the PhD was to check whether I can predict proton chemical shifts in the protein-ligand systems based on the crystal structure. So I had a beautiful crystal structure of this protein-ligand system, where we could see that the ligand interacts, creates weak hydrogen bonds like CHO hydrogen bonds, as well as multiple CH pi interactions with other residues. And what I did, I used DFT, I used quantum chemistry based methods to calculate the chemical shift, and I chose the ligand and the interacting residues, not the whole protein, because that would be too expensive for quantum calculations. So basically what you can see here, but all together in one system. And I compared my calculated chemical shieldings with experimental chemical shifts for each proton, and we can see a quite nice correlation, and the RMSE around 0.7 ppm. And in the second plot, you can see the error, so the difference between predicted and experimental chemical shifts on each atom. And what you can see here, we see a really huge error on the first proton, which you can see here, that it's the one involved in CHO weak hydrogen bond. So first we thought that maybe it's because the DFT method that we use is wrong. So I did benchmarking using other methods, but the error was always there. So our hypothesis was that maybe the X-ray structure is not perfect, and to be closer to solution state NMR representation, I performed a molecular dynamics simulation of this system using classical force fields. And then on the top of that, I did clustering, and on the ensemble of frames. Sorry? Okay. And based on the ensemble of frames from this MD simulation, I calculated chemical shifts again on the same subsystem, so ligand and interacting residues. And I took the average from many frames from MD to see how the accuracy changes. And we can see that RMSE improved when I take the MD ensemble compared to X-ray structure. And we can see that the error on the proton number 1 reduced significantly. So then let's look at the structure. We can see, and here I also plotted something which is called non-covalent interactions index, where a purple color means a strong non-covalent interaction. So, for example, classical hydrogen bonds, and a blue, green is like weak non-covalent interactions, such as CH pi, and red is repulsion. So here, between the proton number 1 and the oxygen on the protein, we can see a blue disk that represents a weak hydrogen bond, CHO. However, when we look at the frames from the classical MD ensemble, we can see that this hydrogen bond is broken, and there are only some weak interactions there. So the message is that based on these calculations, we can see that probably because a classical MD ensemble has a better agreement with experiment, we can assume that probably this is the real situation in solution, that this hydrogen bond is not as strong as in the crystal structure. However, what we also observed here was that the error on the proton number 2 actually increased in the classical MD compared to the X-ray structure. And to deal with that, I went beyond the classical dynamics, and I performed much more expensive QMMM molecular dynamics simulations using, so it means that I selected the QM region of my system, which was what is interesting for us. So the ligand and the interacting residues, they were in the QM part, and the rest of the system was treated with the classical force fields like in the previous simulation. And then again, I chose many frames, and I calculated chemical shift for them, and I took the average. And here we can see the comparison between classical MD versus QMMM, and we can see that RMSE decreased. And here also the error on the proton number 2 decreased, and all of the chemical shift errors on each atom are below 0.5 ppm. And again, looking at the non-covalent interactions index, we can see that during a classical MD simulation, we had a weak hydrogen bond, CHO, between the proton number 2 and the oxygen on the protein side. However, this oxygen during a QMMMD simulation, so higher level of theory, was involved in another interaction on the protein side. So we can see, based on the perfect agreement with experiment, that QMMD ensemble gives the best picture of the real protein-ligand interaction in solution. So we were able to refine the crystal structure to understand the interactions in solution better. And also we were able to identify the artifact of classical MD by this way. And to sum up, So you can see first in the scheme that first I used the crystal structure of protein-ligand system, and I calculated chemical shift only using that and a single frame. And then classical MD followed by clustering and chemical shifts prediction for the whole ensemble and the average, and then QMMM. And the summary is that proton chemical shifts are sensitive probes of non-covalent interactions, and in this case for protein-ligand interfaces. And using molecular dynamics, we can we can refine the X-ray structure using chemical shift prediction, and that QMMM molecular dynamics can even overcome the force field limitations. And this brings me to the second paper, and the second protein-ligand system of study, and this is streptavidin biotin, which is one of the strongest interacting protein-ligand systems. And our goal here was to understand the interactions that lead to this high stability. And we started with the same approach. So we had experimental chemical shifts on the protons in the bound form. And we compared the experimental versus calculated chemical shifts. And in this case, we can see that there are no errors above 0.5 ppm. So we already, from the X-ray, we already got a reliable representation of the system. So we could further analyze X-ray structure with no need for MD simulation. So the next step was to analyze the chemical bonding in these systems, as it was easy to study because we could use only the X-ray. And for that, I used extended transition state natural orbitals for chemical valence approach that is under development at the Jagiellonian University in Krakow, where I did my master thesis. So we use this method to understand better the interactions of the ligand with the binding pocket. And according to this method, the total interaction energy between the ligand and the protein can be decomposed into physically meaningful contributions. The first contribution is electrostatic, which can be favorable or unfavorable, depending on the charge. And then we have orbital contribution that covers the charge transfer, so the interaction energy between occupied orbitals of the first fragment with unoccupied orbitals of the second, and vice versa, as well as the polarization. So here interactions energy between occupied and unoccupied orbitals within each fragment. And here we can further decompose it into energy contributions that are significant for that are chemically meaningful, and we can also visualize their differential density that you will see later. So for example, imagine if we have, I know, multiple chemical bonds, then we can distinguish sigma pi orbitals. And then we have also London dispersion contribution, which is always favorable. And this is based on semi-empirical calculations. And we also have Pauli repulsion, which is always unfavorable. So it's Pauli repulsion between occupied orbitals of both fragments. And I built 3 different systems where I considered the biotin and the interacting residues. And the first one is the biotin and all the interacting residues within 5 angstroms. The second one is biotin with only residues interacting with the head of the biotin, so with urido subpocket. And the third one is biotin interacting with the tail subpocket. And then when we look at the energy calculations, we can see that the black dots means the total energy. And we can see that first, looking at the whole pocket, the contributions to the total energy are more or less equally important, but the main one is electrostatics. And for example, for the tail subpockets, here we can see that electrostatic and dispersion are equally important. And we can also see that the tail subpockets gives more raise to the total interaction energy than the urido subpocket. And this is the differential density of the system. So it's the total energy of the system, total density of the system minus the density of fragments. And the blue color means the inflow of the electron density, and the red color means outflow. So for example, you can see here a strong hydrogen bond with a carboxylate moiety, NHO hydrogen bond. You can see a lot of hydrogen bonds around the head of the biotin, strong hydrogen bonds, but also weaker CHO and OHS non-classical hydrogen bonds. And now I would like to focus on the interactions of biotin only with the tail subpockets, because the interaction of the head of the biotin, they were already studied in the literature. So let's look in the details on the bonding channels of the tail subpocket. So here we can see a lot of, we can see the channels corresponding to sigma contributions of the classical hydrogen bonds with the tail, as well as pi contributions. And further, we can see, here you also have the energy estimations. You can also see a weaker contribution from sigma OHS hydrogen bonds, as well as weaker CHS. So you can see that the sulfur interacts not only with the OH group, but also with carbon H. And then we can also identify a very weak contributions. So the charge transfer corresponding to CH pi interactions. And now I will show you even further fragmentation to fully understand the contribution coming from each amino acid here. So we have the biotin interactions with single residues. And they are ordered from the strongest to the weakest. So first we have a classical hydrogen bonds with glycine and serine. And we can see that they are dominated by electrostatic forces, which is typical for classical hydrogen bonds. Then we have weaker CHS, CHO, OHS interactions. And finally, we have the weakest types of interactions, which are CH pi and even CHAC, that are quite controversial in the literature, as you know, they when we look at CHAC, it looks like it's the same charge, so we assume it should be repulsive, but they are actually found to be attractive. And you can see that all of these interactions are dominated by dispersion. However, we can also see the contribution from the orbital interactions, and you can see, for example, here, a beautiful charge transfer from CHs to the pi ring system. And we can see that the contribution of all these weak interactions is 44% to the overall stability of the system. And this is important because these interactions, they are often rather associated with hydrophobic effect. However, we can see that they are, because there are so many of them, because of their multiplicity, their overall contribution is meaningful. And the next experimental step was that we measured not only chemical shifts on the ligand, but also chemical shift perturbations. And we identified, actually it was Sarah, our master student, Sarah identified strong chemical shift perturbations, especially on the protons that interact with tryptophan. So we can see the chemical shift of them even below 0 ppm, and the chemical shift reaching even 3.2 ppms. And our goal was to understand why these shifts are so strongly shielded, and does it correlate with the strength of the hydrogen bond. So we know that when we have a typical chemical shift, when we have a proton interacting with, for example, the oxygen, we usually observe a de-shielding due to hydrogen formation compared to the vacuum. So we can see a higher value of chemical shifts. However, when we have the aromatic ring round, we usually observe shielding, so very low ppm values, which we associate with ring current anisotropy. However, the question is, because it's also a weak type of hydrogen bond, should we also observe a de-shielding due to weak hydrogen bond formation? And to understand that, I found a method to calculate this both fragments. So I used the very simplified system, only the biotin with one tryptophan, and the first part was to calculate the environmental shielding. And how does it work? So I have only my hydrogen bond acceptor, so only the tryptophan, and I remove the biotin from my system, and I calculate the chemical shielding in the place where my proton would be. So we call it a dummy atom. So there is no atom there. We just compute using quantum mechanics the environmental effect. And then to calculate the de-shielding due to hydrogen bond formation, we need first to calculate the chemical shift of the proton of interest in the whole system, donor and acceptor, minus the chemical shift on only the hydrogen bond donor, minus the environmental shielding. And this method was applied for many model systems in this paper. So I use that method. As I said, this method, here I will present the results for these two protons. And we can see that environmental shielding dominates over the shielding due to hydrogen bond, as we expected, because these hydrogen bonds are very weak. But we can still see that this effect is here. And this is because of the charge transfer that we visualized before. And here I also plotted the experimental chemical shift perturbation. However, we cannot quantitatively compare it with other bars. But what I wanted to say was that the bigger chemical shift perturbation doesn't mean the stronger interaction, because here we can see that on the proton H prime, the de-shielding due to hydrogen bond so the interaction is stronger than on the other proton. However, the chemical shift perturbation is bigger on the proton edge. So this is a misinterpretation. And what we also observed and found interesting was the same calculations on proton F, which only creates CHA-C interactions. And here we could also see a very tiny deshielding due to hydrogen bond, 0.12 ppm due to hydrogen bond formation. And to sum up, we show that the interactions in biotin-streptavidin system are dominated by electrostatic, followed by orbital and dispersion contributions, and that weak interactions such as CH-pi, CHAC, OHS contribute 44% to the complex stability, and that huge chemical shift perturbations that we observed are mainly attributed to a strong ring current effects with small contributions coming from weak hydrogen bonds. And the outlook here is that from both projects, we can say that validating calculated chemical shifts with experimental chemical shifts has a potential for refining biomolecular structures, protein-ligand systems, not only based on X-ray structures, as I did, but also for molecular docking when we don't have the X-ray structure and we rely only on AlphaFold. And we believe that this could support early stage drug discovery. And this brings me to the last paper, which is the machine learning predictor for chemical shifts for protein side chains chemical shifts. So during my PhD, I had the chance to do a secondment at UC Berkeley, where I meet Jerry, and Jerry was the developer of UCB Shift for the backbone, and we together we decided that it would be great if someone extends UCB Shift to work also for side chains, and this was my job there. So to introduce you UCB Shift, it has two modules: UCB Shift X module, which is a machine learning part, and it relies on random forest regression and engineered features that I will show in the next slide. And UCB Shift Y module, which is inspired by Shift X2 predictor that maybe some of you know that work with proteins. But Shift X2 relies only on sequence alignment. However, UCB Shift also uses structural alignment, because imagine that sometimes the sequence of the protein can be very similar, however, the structure can be different, and the other way around. And this UCB Shift Y module, there is no machine learning here. It's just a simple transfer of chemical shifts from the database if we find enough similarity. And let me show you the features. And this is the slide I got from Jerry. The features that we used from the protein's geometry, from X-ray structures, are, for example, a backbone and side chain dihedral angles of the proteins, residue types and secondary structure of the proteins, the geometries and energy of hydrogen bonds, so both angles and distances, ring current effects, and here accessible surface area, which is the surface area of the molecule that is accessible to the solvent, and the half surface exposure, which measures how deeply amino acids are in a protein. And we also have features that correspond to polynomial transformations of the presented features, because, for example, it is known from the literature that it's not a distance of the hydrogen bond, let's say, that directly corresponds to chemical shifts, but it's rather distance to the power of 2 or 3 that better corresponds. So we also have these transformed features. And this is the design of the algorithm. So the user starts with the PDB structure. And let's go through the module Y first. So the blue one. So first, we check in the dataset if there is a similar first sequence and then structure to the protein of the user. And if we do the alignment, and if there is enough similarity, we transfer the chemical shift, and this is UCB shift Y prediction. And then UCB shift X is that we extract the geometrical features from our geometry. And then there is the first extra tree regression to get intermediate predictions, and it's followed by random forest regression. And then from that, we get a UCB shift X prediction. But there is also UCB shift option that takes both. So takes the prediction from UCB shift X together with UCB shift Y. And there is a random forest regressor that combines both of this information. So we have 3 options. And UCB shift 2, so UCB shift for sidechains, was trained on 851 PDB structures, and the test set that the results I will show you on the next slide had 200 structures. So here I show you the comparison of UCB shift to 0, so only the sidechain chemical shifts, compared to shift X2, that is another predictor that covers side chains as well. And here you can see the sidechain chemical shifts average over each amino acids, here for carbon and here for proton. And we can see the overall improvement in both cases. And in particular, you can see on the histidine huge improvement, which is probably associated with the fact that how we handle the protonation states by new features and by different protonation algorithm. And then we can also see a significant improvement on the tyrosine on carbon, which is probably because we had a much bigger training set that covered quaternary carbon atoms. And here I show you the correlation coefficient. So we look at the predicted chemical shifts versus observed chemical shifts from shift X2 and UCB shift. And you can see that correlation coefficient when we plot, for example, all of the carbon gamma chemical shifts, and this is how it was done in the shift X2 paper, we can see a perfect correlation coefficient 1. However, when we look into the details, so I colored each amino acid differently. You can see that there are no variations from the random coils. So there is no correlation within each amino acid type. And here we can see more of the correlation in the UCB shift. However, to see it better, we subtracted chemical shifts from the random coil references. So we look at only at the variations of the chemical shifts from the unique protein structures. Without the random coil references. And here you can see how the correlation coefficient drops from 1 to 0.18, and here as well, but it's a bit better. So Yes, you can see that actually Shift X2 is not able to predict any of these variations from the random coil. It only tells us more or less for something that we know, right? Because we all know random coils for each atom type, or we can check that. However, what we want to predict is what is the effect of the protein structure on the chemical shifts. So we want to have R1 in this case, but R 0.7 in this case is also not that bad. We can see that it's doing something. And then here is the example of an interesting question I got from the reviewers. The reviewer was wondering if UCB shift works better, maybe only because we have a bigger training set. So he asked us to train and test UCB shift on Shift X2 data. And here we can see that still UCB shift can beat Shift X2. So it means that it's not only the data set, but the algorithm is better by itself. And here I also show UCB shift without testing mode. So it means that if we have 100% similarity, we also include this prediction. And this is also what Shift X2 does. And now let's look at input feature importance. So yes, so this is about the geometrical features I showed you before. And what we could see was first that UCB shift Y dominates the R2 regressor. So when we have both X and Y module, the UCB shift Y feature is more important. And then when we look at R1 regressor, so without UCB shift Y module, we can see for here also for backbone atoms that dihedral angle feature dominates, followed by secondary structure. However, for the side chain, we can see also the dominance of dihedral angle feature, but it's followed by hydrogen bonds, as well as accessible surface area and a half surface exposure, as we could expect, because they are side chains. But what was interesting to see was that ring current effect is super important for side chain protons, much more than for backbone. And for some of the proton types, it was even over 60%. So we decided to look at this feature deeper and look only at the very extreme examples with the largest shielding we found. So I took the subset containing hydrogen atoms with the lowest chemical shifts, but also I looked at the geometries of the CH pi contacts of them to find those with the most perfect contact for the CH pi. So what you can see here are the differences between predicted versus experimental H beta chemical shifts, because they were the easiest to analyze in this case. And these are only, so the experimental chemical shifts, they were all below 1 ppm. And what we can see is that on the left side, we have the cases with the strongest ring current effect, so the lowest ppm values, and on the right side, the highest. And you can see for the Shift X2 that on the left side, we have more blue, so we have more errors, bigger error on the protons, on the most extreme protons with the strongest ring current effect. So this is because Shift X2 is not able to handle this CH pi interactions properly, and then similar Shift X plus module of Shift X2. And then we can see that also the UCB shift results, they perform overall better. And the best model in this case is UCB shift X. So UCB shift relying only on the geometrical features. And here you can see that there is no that the errors is quite equally distributed. So it can also handle the most difficult cases much better than Shift X2. And then I also made a test if it is actually the ring current feature that causes this. And yes, so if I run UCB Shift X with turning off the ring current feature, we can see that the error grows significantly. And as I said, UCB Shift X exhibits the highest level of performance here. And yes, and this is all about UCB Shift. However, I have also a little outlook. At the end of my PhD, together with my organic chemistry friend, Giorgia Toscano, we decided to apply for a new ideas grant from our PhD program. And we applied for the project titled Automating a Metal Assignment in Protein NMR by Machine Learning Chemical Shift Predictions and Isotope Labeling. And that was at the very end of the PhD, but we recently, at the end of last year, managed to publish the paper with the big help of our colleagues that helped us a lot with the NMR part. And what we did, so Georgia is an organic chemist, so she designed the precursors that were needed for this project, and other colleagues designed the new pulse sequence for a metal to backbone coherence transfer. And my part was to use UCB shift for automated assignment of chemical shifts here. So I would only like to discuss this part now shortly as the follow-up of what we can do with UCB shift 2.0. So from the experiment they performed, we got chemical shift for leucines and valines. So all of them that are in purple are those that we can see from the experiment. So I is leucine and I+1 is the next residue in the sequence. So we get all of this chemical shift except of C gamma, because according to this labeling scheme, we cannot see this chemical shift, and for valines we cannot see C beta. And what algorithm did was, yes, we had a PDB structure, either X-ray or AlphaFold, and then we performed UCB shift to 0 calculations, and we got this chemical shift set for all the leucines in the protein sequence. And then we had also experimental values, right? So all of these chemical shifts for all the leucines. And then I calculate RMSD between each pair. So each leucine in the sequence versus each experimental leucine. And this was weighted RMSD, so the weights were bigger for more significant protons and smaller for example, nitrogen. And then by matching RMSD from the lowest to the highest, we were able to do the assignment, both for leucines and for valines. And the performance was for valines on the test set of 8 proteins. It was 95% and 86% for leucines. And that brings me to the final slide, to the acknowledgements, which is the timeline of my PhD journey that started in Graz because of the collaboration with the Medical University of Graz with Pedro Sanchez Murcia, that was my co-supervisor on the computational chemistry side. And then I visited UC Berkeley. Teresa had Gordon Lab, where we came up with UCB Shift to 0. Then I moved to Vienna to my main supervisor's lab, Robert Conrad, and then I joined the project with Georgia, where we created the last paper that I showed you. And after that, I was working in AI Affinity, a biotech startup. And then I would also like to acknowledge my main collaborators, which is the MagLab from Vienna, Boehringer Ingelheim, and the group where I did my master's and bachelor's, the group of Mariusz Mitorai from Jagiellonian University. And also thank you for your attention.
Thank you very much, Aleksandra, for your great talk. And we are open for the discussion. So if anyone wants to ask questions, please turn on your microphone and, if possible, camera and ask the question. I strongly encourage students to ask questions. Do we have any volunteers for questions? Oh, Justyna, yes, please.
Hello, guys. So, Alexander, very, very nice talk. Super excited to see that. So I have one question for you. And currently we can generate a lot of pretty good docking poses with AlphaFold 3 and with Boltz tool, yeah? Do you think we are there with the chemical shift predictions to use them to rank the correct Boltz 2 models, or your error bars are too far? Just to put in perspective, Shifty 2 was just not working at all. So I'm curious how your, especially ILV approach would perform in your opinion.
But what experimental data do you have?
So I would have the just chemical shift perturbations.
Yes, but on the protein side?
So I will have AlphaFold model. So the PDB file from AlphaFold or from
Yes,
Boltz 2.
but the chemical shifts on the protein, right?
Yes. Yes.
So Shift X2 will not work, and
Yeah, I
UCB
know, I
shift will
tried.
not work because they are only trained on protein systems, so not on protein-ligand systems. So they will not see the effect of the ligands at all. So you would have to use actually the ab initio quantum shifts predictions to do this, to deal with that, because there are no there is no any machine learning predictor that would work faster. And I think recently, I think 2 weeks ago, one professor contacted me that he would actually like to build this kind of model based on our papers that would be trained on that. First he would have to prepare a data set, because we don't have experimental data set that would cover protein ligand systems. There is not enough data, especially on the ligand side. So first he would like to design the data set, the computational data set. So he would perform QM calculations for the huge data set, and then he would train the model to do that. So that's the idea. And currently, yes, you can only do that with QM calculations. And yes, I think it's definitely worth trying, or if you want, I can also help you. And I think I would recommend the approach, so the DFT methods. I don't know how much familiar you are, but the ones I used in the paper, because this is based on a lot of benchmarkings. But I believe that could work. I also saw some papers, I think 1 or 2, where people were trying to do the docking based on chemical shift perturbations and based on or only chemical shifts, by comparison of QM versus experiment.
Thank you so much.
Do we have any other questions? Let's give people a moment. Students are very encouraged, are reminded about the competition. Okay, they are shy today. Oh, no. Krzysztof, yes. However, not student. Krzysztof, I cannot hear you.
Now?
Yes.
Yes.
Better? Yeah, so thanks for the nice talk. I have a question about this interesting CHAC interaction. Is there any hypothesis explaining why it's attractive? Because you say it's controversial, but there must be some explanation proposed, no?
I don't follow this literature anymore. It was kind of part of my master thesis, but at that time, I think there were not really experimental results. They were rather, quantum-based speculations about the attraction there.
Okay.
And I think I heard once a talk where someone based on some, I think, IR spectroscopy studies claim that they are actually repulsive. But, when we have, for example, I think that was graphene. So where we have a lot of CH, when we have a graphene diamer where we have a lot of CH contact, we can see that when we increase the layers, they interact, stronger and stronger. And the only explanation is the corporation of CHAC interactions. So there are many speculations like that.
Okay, thank you.
Okay. I think we still have time for one question if anyone is interested. Okay. Looks like the question pool is finished. So I will start now, introduce the next lecture. So the next seminar will be in 2 weeks. It will be given by Agnieszka Skorupa from Maria Skłodowska-Curie National Research Institute of Oncology in Gliwice, and will be titled NMR-based metabolomics in the studies of gynecological cancer. After that, we'll have 3 more talks, as you can see. So thank you everyone for joining us, and see you in 2 weeks. Aleksandra, if you could stay for a little longer, I would be very grateful. So thank you very much for everyone.
Okay, sure.
Thank you. Bye.